Well, thank you so much, Dr. Kuskoff. That was an amazing presentation. Want to appreciate and send appreciation to your lab and also EHE Rare Cancer Charity UK for helping support that. I know as a patient, I can speak to say that when I was reviewing your presentation, a lot of it was over my head. And I am so happy about that because I'm so happy that we have wonderful minds working on EHE. So thank you very, very much. So next up, I am excited to introduce Dr. John Lamar. He is with us today to share identification of negative regulators of the TAS-CAMTA1 fusion protein. We are excited to have Dr. Lamar with Albany Medical College with us today and can't wait to learn more about this important topic. And please welcome Dr. Lamar.
Hi, and thank you very much to the organizers for inviting me. It's a really great workshop, and I'm happy to be involved. And so, as you heard, I'm going to tell you a little bit about our project we've been working on, looking to identify negative regulators of the TAS-CAMTA fusion protein. And so, as we've heard this morning, roughly 90% of EHE cases will have this WWTR1 or TAS-CAMTA1 fusion protein, with the other 10% having the closely related YAP protein fused to TF3. And we know from work from Brian Rubin's lab that this TAS-CAMTA1 fusion protein is oncogenic and that it drives a TAS-TEAD-like transcriptional program. So what that means is it turns on a lot of target genes that are very similar to the target genes that the wild-type TAS protein turns on when partnered with another family of transcription factors called the TEADs. What's really interesting to me about this disease is this fusion protein isn't found in any other type of cancer. Yet, if you look at other cancers, you see activation of TAS, which would presumably drive this same transcriptional program or a similar one, playing a really causal role in many cancer types. And so it seems like this fusion protein, in combination with the unique cell of origin in EHE, is really creating a situation where this disease will develop. And I think more importantly for what my lab is interested in, it also suggests that if we could figure out a way to inhibit this fusion protein, it can really potentially be an effective treatment for many patients, since such a high percentage have this protein in them. Their tumors are likely dependent upon it. So I have been studying YAP and TAS for over 15 years now, and when I first learned about this disease through Brian Rubin, and I looked at the fusion protein itself, I was immediately struck by the fact that it still retains a lot of the same negative regulatory sites that the wild-type TAS should retain. And so this includes the LATS phosphorylation sites that allow the Hippo pathway to repress TAS. It includes two phosphorylation sites that are important in the GSK3 beta mediated degradation of TAS. And it includes this serine 51, which is really important for TAS's ability to bind to the TEAD family of transcription factors. And so this is also negatively regulated by some pathways for TAS. And then a subset of the translocations that have been sequenced seem to contain an intact WW domain. Many do not, but some do. And there's a number of negative regulators that bind to the TAS WW domain. And so it looks like at face value, this protein should be regulated by many of the same things that TAS is. However, we know from work from Brian that even though these LATS phosphorylation sites are retained in the fusion and they do get phosphorylated in a Hippo-dependent manner, the protein seems to be largely insensitive to this, and so it kind of ignores the phosphorylation event, and that's probably because the CAMTA portion of the protein carries the protein into the nucleus despite phosphorylation. But we also know from Brian that you do need this serine 51 to be present in TAS, and if he mutated that, they saw that it lost the oncogenic activity. And so regulatory mechanisms that influence this serine should also influence TAS-CAMTA1. And we do not know about these serines now, and that's one of the things that we're asking about. So it does look like at face value to us that this protein should be regulatable in cells. And so this is just a list of data from the YAP-TAS field outlining some proteins that are known to regulate TAS or YAP, which has many of those same domains, in a manner that's independent of the Hippo pathway. And so we predicted that many of these might also be able to repress or regulate the TAS-CAMTA fusion protein. So all the work I'm going to tell you today is from a talented master's student in my lab named Ryan. And so what Ryan did was he set out to set up a model system where we could test whether any of these regulators could repress the function of the TAS-CAMTA fusion protein. And so because Brian had shown previously that TAS-CAMTA1 is oncogenic in NIH3T3 cells, and because we don't have any EHE cell lines to work with at the moment, we set out to make this similar model system to the one that his lab has used. And so using constructs, retroviral constructs that he has given to us kindly, Ryan took NIH3T3 cells and he stably expressed the TAS-CAMTA1 fusion. This is constitutively expressed, unlike the talk you just heard, so it's always on in these cells. And then the idea is to take these cells stably expressing this fusion protein and express in them either the negative regulators or knock down things we predict would be positive regulators. And then we're going to use a series of assays to test how this influences the function of TAS-CAMTA1. And I'll show you some oncogenic activity assays that we have both in vitro and in vivo to look at some of the target genes that we know are regulated by TAS-CAMTA1, and then to look at the transcriptional activity, which we can measure using this YAP-TAS reporter assay. And so just real quickly, the way that this works is it's a vector that has a firefly luciferase gene. So this is the gene that encodes the protein that allows a firefly to light up. And we've put it under the control of a promoter that only responds to TEAD. And so when TEAD is binding this promoter and being in partnering with YAP or TAS, it will turn on lots of expression of this firefly protein, and the cells will emit light when you give it a substrate. And we can measure that as a direct readout for how actively these proteins are transcribing or driving transcription. And since it retains all of the same motifs as the wild type TAS protein that are responsible for this, we predicted that this would be a nice readout for TAS-CAMTA activity as well. And as I'll show you, that is the case. And so we can just transfect this into cells and it will be a nice readout for how active TAS-CAMTA is. So the first thing that Ryan did was he took the viral constructs that Brian gave us, and he infected the NIH3T3 cells with increasing doses of the viral supernatant. And so what this does is it just gives us increasing amounts of the TAS-CAMTA protein being expressed in the cells, which you can see here by Western blot. And what that showed was that we do see a dose-dependent increase in the activity of our reporter assay. And so this tells us that this assay can read out TAS-CAMTA activity, and we can use it for our experiments moving forward. And then Ryan just did this multiple times, so we see that in multiple experiments that the TAS-CAMTA1 activates the activity of this promoter assay that we use. We know this is dependent upon the TEADs, just like Brian had shown in his system, because if you introduce a version of TAS-CAMTA into these cells that cannot bind TEADs because of this mutation we've introduced, it can no longer activate the transcription of that reporter assay. And so this is really just to prove that the system is behaving the way we hope it is. And so now we can start to use this to look for some regulators. Ryan also basically rebuilt this whole thing in another cell line, HEC293 cells. This will just give us an independent way to verify what we find. HEC293 cells are also a little bit more utilizable because they can be easily transfected and manipulated, whereas the 3T3s are a little harder to work with. So we now have two model systems, and again, putting in the TAS-CAMTA1 induces activity of this promoter construct. Ryan also put in a version of TAS-CAMTA1 that lacks that WW domain, and this is important because many of the translocations sequenced in humans do not have an intact WW domain. So the breakpoint is such that this domain is no longer functional. So we wanted to make sure that anything we identify also regulates the WW domain mutated version of TAS-CAMTA1, thereby helping potentially a greater number of people. And so that mutation doesn't affect the transcriptional activity of this protein, but again, mutating the TEAD interaction domain and preventing this thing from binding to its partners, the TEADs, basically blocks its activity entirely. So now we have two different cell line systems that we can use to test for regulators of this protein. We also want to look at the actual oncogenic function of TAS-CAMTA1. And so Ryan turned to an in vitro assay called a foci formation assay. And all this really is you put the cells on a dish and you let them grow until they're really densely packed together. And normal 3T3 cells or fibroblasts will stop growing. It's called contact inhibition. So they don't want to grow anymore once they've reached this confluency. But if a cell is tumorigenic or transformed, as we say, they'll start to grow on top of each other, and they grow out of this culture into these little dots called foci, and you can count these up real easily. And so as you can see, the normal 3T3 cells, as well as the ones in which you've put in a control vector, they don't form these foci. But if you put in the TAS-CAMTA1, you get lots of foci forming, suggesting that this is transforming cells. And again, this is just consistent with what has been reported already. We're just setting up our model system here. So we also wanted an in vivo model system to test this in, so to test whether or not TAS-CAMTA can drive tumor formation, and then whether the things we predict will inhibit it can repress that. And so Ryan just utilized a system that we routinely use in the lab for other cancer types. And so we took the 3T3 cells that are expressing TAS-CAMTA1, and then we stably label them with a fluorescent protein, in this case, a really bright green protein. Or we can put in that same firefly luciferase gene I talked about earlier, and this will allow us to monitor the cells in a mouse that's alive as the tumor grows. And so we can basically track these cells in the mice, both at the end of the experiment and as the tumors are growing. And then the idea will be to put in the negative regulators, and then once those are in there stably, then you can inject these into the mice subcutaneously, and then measure the formation of the tumors that form. And we can also track the spread of this disease to other organs like the lungs or liver if we using the green fluorescent protein. And so just to show you that this works, Ryan did an experiment where he injected mice with the control cells that don't have TAS-CAMTA1 in the left flank, and then the cells that have TAS-CAMTA1 in the right flank. And then, as you can see, the majority of the ones injected with the TAS-CAMTA1 expressing cells made tumors, which after a latency period began to grow very rapidly. And only one mouse had anything measurable or palpable when injected with the control cells, and that tumor didn't grow at all. And so all the black dots are sort of at the bottom here, not showing any growth. So TAS-CAMTA1 drives tumorigenicity in a mouse model as well. And so now we have an in vitro model system and a mouse model system where we can test whether things that negatively regulate TAS-CAMTA1 can disrupt its tumorigenic function. And so with that, Ryan went on to now test some of these negative regulators that we predict might repress the activity of TAS-CAMTA1. And we're going to start with that transcriptional reporter assay that I talked about. And so what Ryan did was he took cells expressing the TAS-CAMTA1 fusion protein or control cells that don't have it, and he put into the TAS-CAMTA1 expressing cells, he transfected in vectors encoding or delivering the genes that will make the proteins for each of these potential negative regulators. And there's a little bit of variability in these 4 assays that he did, but what we saw was that several of these genes were able to repress the activity of the TAS-CAMTA1 relative to its level without any of these vectors. And so we will focus on these ones moving forward a little bit. And just because there's a little bit of variability in this 3T3 system, he also verified that in the 293 model system. And again, you see the TAS-CAMTA1 activates the activity of this reporter assay, but when you transfect in these various proteins that we think should repress it, we do in fact see that it dramatically reduces the activity of TAS-CAMTA1. So it suggests these proteins, when you overexpress them in cells, are able to repress the function of this oncogene. We also did the same experiment in that mutant form of TAS-CAMTA1 that doesn't have an intact WW domain, and we saw that for the most part they also repress that version of the protein, which is good because it means that patients who have a disrupted WW domain might still benefit from these molecular pathways if we can validate them. Okay, so we've decided to move on with most of these proteins, but we've been focusing on AMP kinase, mostly because there are drugs that exist that can actually activate this protein, and they're FDA approved. And so we figured that maybe we could repurpose those once we validate this as a true negative regulator. And so what Ryan's been working on lately is just trying to see whether the endogenous AMP kinase signaling pathway can be modulated to influence TAS-CAMTA1 activity, because in all those previous experiments, we've basically dramatically overexpressed the protein. And so he's working on a drug right now called ICAR. It's a small molecule that can actually activate AMP kinase, and that's what we predict would be repressing TAS-CAMTA1. So those experiments are ongoing, but he has data now with a compound that actually inhibits AMP kinase So this would be the opposite of what we'd want to do in EHE, but it's just to show that the endogenous pathway is working. And so if we treat cells that have TAS-CAMTA1 with this either a control vehicle or increasing doses of this inhibitor of AMP kinase, you see that actually the activity of TAS-CAMTA1 goes up dramatically in the NIH3T3 cells. So it suggests that in those cells, AMP kinase is actually repressing TAS-CAMTA to some extent already, and that the endogenous machinery in the cells can be exploited. And so now we just need to know whether we can activate AMP kinase to repress the levels of TAS-CAMTA1, or the expression of activity of TAS-CAMTA1. And so that's where the project is at the moment. Ryan's, like I said, trying to do some experiments now to show that if we activate the existing AMP kinase in these cells, we can repress TAS-CAMTA1. And we've now started to move forward with checking other readouts as well, so that the genes we know are regulated by TAS-CAMTA1 is another readout for its activity. And Ryan has begun the foci formation assays, and we're about to start the tumor growth assays now. So he's made cells that stably express AMP kinase, and we're going to put those into the mice this week. And so hopefully what we'll find is that this represses the tumorigenic function of this oncogene. Obviously, all this was done in NIH3T3 cells, and so we really would like to have an EHE model system to validate some of these findings. And we're really fortunate to be collaborating with Brian Rubin. He's going to tell you about his mouse model in a couple talks, so I won't steal his thunder, but I'll just say that we're hoping to try to get some of the tumor cells out of that mouse model and start testing to see whether these same regulators we've identified in 3T3s can also repress TAS-CAMTA1 in those models. And then moving forward, we'd like to know how it's doing this, and just based on what AMP kinase is supposed to do to YAP and TAS, this is published work from multiple groups that was summarized here. It's been shown that AMP kinase can influence TAS and YAP through a Hippo pathway or LATS mediated process, and we would predict that this is probably not happening in the fusion, since it seems to be rather insensitive to this wing of the pathway. But we do predict that this is probably the mechanism. So AMP kinase can phosphorylate YAP or TAS on the serine that is essential for its interaction with the TEADs. And that phosphorylation event basically prevents the YAP and TAS from binding to TEADs. And so we're disrupting the interaction of TAS-CAMTA with TEAD, we think, by activating AMP kinase. And so we'd like to confirm that that's the case, and also look at the mechanism for some of the other regulators that we found. So just to wrap up real quickly, I've told you that we've developed a model system that will allow us to identify and test potential regulators of TAS-CAMTA1, both at the transcriptional activity level and its oncogenic function. And we've used this system to identify several proteins that can repress TAS-CAMTA1. And I think what's important about this is that it suggests that this protein, although it seems to be so active in these tumors, it can actually be regulated in many of the same ways that the endogenous TAS protein can. And so now we have to figure out why it's not being regulated in the cancer and see if we can exploit some of these pathways to repress its function. And then we've also optimized this same approach to perform larger RNAi screens to identify additional novel regulators of TAS-CAMTA1, and that's something we're moving forward with in the future. And so with that, I'll just thank Ryan is the one that did most of the work, but my lab is very collaborative, so everybody generally helps out with these things. Nothing that I've talked about today would be possible without our collaboration with Brian Rubin, who's been extremely generous in providing reagents, resources, and input. We're working closely with him on this and other projects now. I also have to thank Guy Weinberg and the Cravat Foundation. So Guy and I partnered to organize this conference that I'll tell you about in a second. And it was through his help that I obtained some of the funding to do this project. And with that, I'll also thank the EHE Foundation, or the EHE Fund, which is a joint effort by the 3 EHE foundations to fund my lab. And so they've come together and provided the funding for this entire project. And then my lab is also funded by some other sources as well. And so I'll just end by saying that Guy Weinberg, myself, and Bob Varelis, we co-organized this workshop that's held in Telluride, Colorado, a very beautiful ski resort. We held it in the summer, so it's a very beautiful hiking resort at that point. And what we do is we try to bring people from the YAP-TAS Hippo community together with EHE researchers with the idea that these people will basically, lend their knowledge to this disease. And so far it's been very effective. We've gotten some really big names in this field to get interested in this. And there are some papers coming down the pipeline that you guys will see, and some joint projects that have now started. And so if there are EHE researchers who have not yet been invited, please let us know. We're happy to have you join, and it's a really, really great experience. And so we're on for June of this year, hopefully in person, but also potentially virtual. So you can email me or Guy to get more details. And with that, I'll stop and take questions, and thank you for your attention.
Well, thank you very much. Very, very exciting work, and it looks like the upcoming months are going to be all the more exciting. You're in the sweet spot at this point.
We hope so, yes.
So, two questions came up right away, both related to potential additional compounds to consider. So the first one asked, with respect to the AMPK effect, have you considered the compound metformin, which I was not familiar with. I quickly looked it up. It's a type 2 diabetes drug.
Yeah, so we did some initial experiments with metformin, and it did not seem to drastically activate or repress in the 3T3 model. However, the caveat there was we borrowed some old metformin from another lab, and so we've ordered that fresh now, and we're definitely testing that. That's on the list as well. Also, at the time that we did those experiments, we didn't have all the reagents to confirm that AMP kinase was being activated. But yeah, that is the commercially available FDA-approved drug that we were sort of thinking about when we pursued AMP kinase, because it does activate that pathway very strongly.
Excellent. So, and
again,
the second one is kind of related.
Yep.
Question here is, adenosine is widely used in inexpensive drugs as a drug. Any thoughts of trying it?
Yeah, it's probably worth trying that as well. We haven't tried that yet. I think we'll probably start with the metformin and see what that does. If for some reason activating the endogenous pathway is not sufficient, we're going to have to figure out why. We do see a slightly greater effect with one of the alpha subunits than the other, and it might be that one has a more dramatic effect. So AMP kinase alpha 1 doesn't activate as strongly as alpha 2, doesn't repress as strongly as alpha 2. And so I think we'll probably try to get to the bottom of that before we expand our drug list, but I think it's definitely worth trying that as well.
Got it. Yeah, and I just received a comment, which is actually relevant to this. It says, we have several patients report metformin brought stability, many of them who are diabetic. So, some people were sort of indirectly receiving it as a therapy for other purposes.
Yeah, and I think one thing that's sort of nice about the idea of metformin is it's not horribly toxic, and people take it for other reasons. And so, again, if we can co-opt existing therapies, that's always going to be much quicker and more effective. And of course, this pathway is responsive to other things too. So, it might suggest that controlling one's diet could also help with EHE management.
Yeah, interesting. With respect to the fact that you've got specific molecular targets that are different and possibly working by different mechanisms, have you considered or plan on looking at a cocktail approach, possibly targeting two or more of them together to get either an additive or synergistic benefit?
Right. So actually, my lab is very interested in that, both in the YAP-TAZ space and the TAZ-CAMTA space. And I'll just show you real quickly. We've developed an approach that's quite complicated, but it allows us to do just that. So we can really use DNA barcodes to label different populations of the TAZ-CAMTA1 expressing cells, and then basically manipulate different groups of those genes in combination, and then inject the mixes into the mice. And then we can use qPCR to basically identify which mixtures were the most effective at inhibiting the growth of the TAZ-CAMTA expressing cells. And so we would start with a genetic approach to try to identify which combinations of those genes, when overexpressed, would give us the greatest effect on growth. And then we would go back and test the drug cocktails in the mouse model as well. And again, all this would ideally be done in an EHE cell line if we can get one of those up and running. But we'll probably still use the fibroblasts now as a way to test them. So yeah, we're definitely thinking about these combinational approaches, mostly because there are lots of ways to turn on and turn off YAP and TAZ. We know that from the YAP-TAZ literature. And so it actually does suggest that we may have to use these combinational approaches to get a complete repression of the protein. But yeah, that's a good idea.
Excellent. Well, thank you so much. We're out of time for the Q&A. Very much appreciate this talk, and we'll be excited to watch your results as they arrive in the upcoming months and years.
Thank you.